Okay, and in 2 minutes, our discussion will be online, and there will be audience from several platforms.
Mm-hmm.
Okay, you should be able to see my screen.
Yes.
Yeah. Yes. Yeah.
It's evening in China. It's 7 p.m. So I will say good evening, everyone. I know it's your morning or noon.
Yeah.
It's lunch now. Yes, for me it's 12. For Georg it's 11.
Yeah.
But 7 o'clock. That's a common time for presentations over there.
Yeah, because most people of course not a common time. Normally we have daytime, but for cross-country communication, normally we choose the evening.
Mm.
So Li Qing, let us know when it's on.
We have 15 minutes to go. 15 seconds, sorry. Sorry for that. Okay. So
We have 15 minutes.
5, 4, 3, 2, 1, go.
Hello, everyone. Good evening. It's a great pleasure to have our special seminar about the Understudied Proteins Initiative, 为充分研究的蛋白质计划. Today we are very glad to have two speakers, Professor Julie Rapsy-Silber from University of Edinburgh and Professor George Kustarcher. Sorry if I did not pronounce your name very well, from University of Edinburgh. So I will first introduce two speakers, followed by a short advertisement of our PhD program. So Professor Julie Rapsy-Silber, he is a Wellcome Senior Research Fellow, Professor of Proteomics in the University of Edinburgh, and a Professor of Bioanalytics in a Technological University Berlin. His group develops tools to study the function, location, interactions, and the structure of proteins in cells. Most of the work involves cross-linking, mass spectrometry, machine learning, and software development. So his CV is shown here. I believe all of you have seen his CV in a poster. Our second speaker is Professor George Kustarcher. He is a group leader and MRC Career Development Fellow at the Institute for Quantitative Biology, Biochemistry, and Biotechnology in Edinburgh. He studies molecular biology at the University of Salzburg, Austria, and obtained a PhD from EMBO in Heidelberg, Germany, working on epigenetics in the lab of Andreas Labdunner. So his detailed CV is here. So aim of his lab is to understand how cells regulate protein levels and how these processes are disrupted in cancer cells. They address this question by combining proteomics, transcriptomics, and computational approaches, including machine learning. So I know these two speakers through a joint initiative, Understudied Protein Initiative. Today I'm very keen to know what's happening and what's the current state and what we can do in the next few months. So myself is a PI at Westlake University. I'm working on proteomics and clinical application. Lastly, advertisement of our PhD program in Chinese. Sorry, just a few seconds.我们西湖大学呢，最近呢正在进行下一次博士生的招生，本科生、硕士生都可以直接的申请，报名的时间 要求都在这儿，我们只申请考核制谢谢。Okay, I finished my presentation and a very short introduction. Let's welcome Jury. Are you going to be the first speaker?
Thanks a lot for the very kind introduction and for inviting us to share this exciting initiative and opportunity with us. I found it's part of the internationalization of science, which is an exciting undertaking and really greatly welcomed. That we all stumble over each other's names. It's great. When I have to introduce you, I also always a bit uncomfortable because I think I massacre your name. And it's good to see that we all have the same problems with each other. So
yeah,
good. But Georg will actually open the floor.
Yeah.
So I hand over to you, Georg.
Yeah. Hi. So yeah. Thanks also for inviting us and giving us this opportunity. So Yuri and I were part of a team of people who are working on starting this understudied proteins initiative. And so over the next half hour or so, our goal is to show a little bit of what the problem is and why it matters and how we could address it, and also how others could get involved in this initiative in the future. So I want to start with a joke. There is a drunk man looking for something under a streetlight, and a policeman passes by and asks him, "What are you looking for?" And he says, "I'm looking for my wallet." And so the policeman starts helping him to search. But after a while, and they still haven't found it, the policeman asks, "Are you sure this is where you lost your wallet?" And the drunk man says, "No, I lost it in the park, but this is just where the light is." And so this, in various forms, this anecdote has appeared over centuries in many cultures, and it refers to a type of observational bias that also affects scientific research. It's basically this idea that the best place to look for something isn't always the best place where that thing can be found. And it's referred to as the streetlight effect. It matters in biology, among other things, because of the existence of understudied proteins. And this is the problem that we're facing. So there is a massive bias at the moment in the functional characterization of the human proteome. And it's been documented in many different publications. A few are highlighted down here. But just to give you a quick overview. So one example is P53. That's the most popular protein in the human genome. There are on average 2 publications every single day that come out on P53. But at the same time, there are thousands of other human proteins on which there are no or barely any publications at all. And more generally put, about 95% of all publications in the life sciences focus on only 5,000 human proteins at the moment, whereas, as you probably know, we have around 20,000 or at least 20,000 proteins. So that clearly shows that we're focusing on only a small part of the proteome that exists. It was believed that when the human genome was sequenced, that this would stop or mitigate this bias. But in 2011, so 10 years after the genome was sequenced, 75% of all publications still focused on genes that were already known before the genome sequence was published. And so basically, people have not really started working on newly discovered genes. They kept working on things they were already working on before. And that's just for the human proteome. The problem is, of course, much worse in other species like yeast or Drosophila, and even worse in non-model organisms, which can be very important because that includes, for example, pathogenic bacteria, fungi, and so on. Now, some people might say perhaps some proteins are just not studied as much because they're not very important. However, we now know from large-scale experiments such as genome-wide association studies and other like data-driven big projects, we know that this just is not the case. So again, I just want to give you a few examples for evidence that understudied proteins are not less important than proteins that are well studied. So for example, a few years ago, a paper looked at genes that were essential for proliferation in a human cell line. And what they found is that 18% of those genes were uncharacterized still. And even more extreme, when Craig Venter's lab published the synthetic minimal bacterium, so an organism whose genome had been engineered to carry the lowest number of genes possible for it to survive, they found that, or they reported that 31% of the genes that this minimal organism needed were still without a known biological function. And more recently, we've seen a similar thing happening with COVID. So there was genome-wide association studies that identified human genes, host genes, that somehow were important for disease progression or severity and so on. But more than half of these genes were never actually followed up in targeted studies or never addressed in targeted studies. Understudied proteins may also be important or are definitely important clinically. For example, we know that many rare diseases involve proteins that are poorly understood, but rare diseases collectively are not rare at all. And even in very common diseases such as cancer and neurodevelopmental disorders, we find more and more that it's rare genetic variants that are important for driving the disease, and these are often also understudied. And the Illuminating the Druggable Genome Consortium has reported that out of all proteins that they consider to be druggable in principle, only 5 to 10% are actually targeted by existing FDA-approved drugs. So there is definitely a level of understudiedness when it comes to clinical applications. And lastly, I just want to say that this is probably just the tip of an iceberg, because we are still discovering more and more genes every day. Just in July, a big consortium consisting of Ensemble and UniProt and others reported the identification of 7,000 new human genes, and those had previously not been detected because they are so small. So they are mainly encoded by what's known as SMORFs, or small open reading frames, and they encode very small microproteins. And being newly discovered, of course, they don't have an annotated function yet. So the streetlight effect on understudied proteins means that we have a problem in the annotation, that some proteins are very well known and some proteins are just never being studied. What could be the reasons for that, for this inequality in the annotation states? So they basically fall into at least two broad categories: those that have to do with practical considerations and those that have to do more with the way science operates in terms of research culture. In terms of practical considerations, it can be things very simple, like the properties, the physical chemical properties of a protein. For example, how abundant a protein is will of course determine how easily you can detect it, for instance, by mass spectrometry. How soluble it is means that a more soluble protein is easier to manipulate biochemically, and so generally abundant soluble proteins are better studied. And one factor that is particularly important, it seems, for or correlated with how well known the function of a protein is actually its size. So this figure here is from a paper that Yuri and I published a few years ago, where we looked at the UniProt annotation score, which is a measure to determine the annotation state in UniProt. 5 means a protein is very well studied. 1 means there is nothing known about this protein. And when you look at two groups of proteins, those that are larger than 15 kilodaltons and those that are smaller, you can see that the small proteins, they make up a very small fraction of the well-known proteins, but they make up almost 40% of the fraction of the very poorly annotated proteins. So size is a clear factor here. Another factor that matters are lab conditions, because as in an effort in science, of course, to standardize our results and to make them more reproducible, we tend to work with very similar or same cell lines in the same cell culture media and so on, to be able to compare results. But that also means that any protein that is not expressed or active under those standardized or limited conditions will often not be detected or studied. Third, the available knowledge is important. If you have a phenotype of a knockout, for instance, or if you know about molecular interactions, protein-protein interactions, etc., it's more likely that you can design an experiment to address what a protein might be doing. And the same goes for the availability of tools. If you already have, for example, an antibody to detect a protein or a plasmid to overexpress it or so, then that really facilitates the way you can, how easily you can address a protein, and that means such proteins are studied in more depth or more often. And the second set of considerations for why some proteins are more studied than others has more to do with the way our science culture or research culture works. So when you look at popular genes or fields, in the sense that more people are working on those genes or fields, then generally these will generate more citations. That means higher impact publications, which then means more funding and more generally academic success. So that's a clear incentive at the moment, often for fields to become larger and larger, and more people to work on in one field rather than starting something completely out there. And a last factor that is often overlooked is the fact that funding agencies often, over the last decades, required research projects to be driven by hypotheses rather than questions. And that, in the context of understudied proteins, is a problem, because if you know nothing at all about the protein, you can't formulate a hypothesis, and that sort of makes it much harder to start working on a completely undescribed protein. And this is just summarized in this simple graphic here. If you have data and tools and publications, then you facilitate working on a protein, and that will allow people to determine its functions relatively easily. But if you know nothing about the protein, then you don't have a starting point, and you never get off the ground with working on this with mechanistic investigations. And that's where the Understudied Proteins initiative comes in, because the idea for us would be to provide a basic characterization of all human proteins, or most, in a way that will allow future scientists to start investigating them and reduce the annotation inequality. And I will hand over to Yuri now to show you more about how about the initiative itself.
Great. Thanks a lot, Georg. So now that you hopefully understand at least the scale and the importance of the understudied proteins challenge, the question is, what can you do and what can we as a group do? So the very first thing is, you could take your mobile phone and follow this link. It will get you to a survey, which you don't have to fill in right now. But this survey should help us to solve a number of questions, actually, that you may even have yourself right now. Because when Georg spoke about understudied proteins, what is that actually? When is a protein understudied, and when is a protein studied? When is it sufficiently studied or insufficiently? It's while we all may have an intuitive understanding of that, we need a kind of like a qualitative and quantitative understanding on a scientific way what that is. And there each of us may in fact come to a different definition, and we would like to have the definition of science, like science as a community. And for that, we need you, because it's only when we all work together on that we can come to kind of like a canonical view on how to make that distinction. So the first question we would like to know is, what is actually the minimal information and tools for a protein that we need for it to become tractable scientifically, to be able to formulate hypotheses, to start working on a protein as a molecular biologist in the laboratory as such. The second question, that was tool or information centered. The second question is this definition, what is actually an uncharacterized protein? So that we can quantify how many proteins are in fact in the moment falling under this. There were a couple of numbers that Georg told you. They were coming from publications. Each of these publications used their own definition though. And we would like to come to not an ad hoc definition, but one that we can maybe all accept then. And not unimportantly, if we try to solve this issue and not solve the issue of undercharacterized proteins and provide initial information, where should we place that initial information for other biologists to find it? And for that, we want to know, where do people look when they investigate what is already known about the protein, because then we would place new information rather there than put it somewhere in a hidden place. It's not, or maybe we need to make a new structure, but maybe we don't need to make a new structure where we place our new information, where we can just place it in the already existing structures. If there's a good go-to place where people tend to go, I mean, maybe it's UniProt, maybe it's some other place, maybe it's Wikipedia. Again, each of us may have their own idea what it is, but we would like to systematically explore where thousands of people are going to find that information. So if you're going to the survey page, what you will encounter is you will actually get a random of these 20,000 proteins, and you will be asked, according to your own definition, whatever it is, how well annotated is this protein? Is it very poorly annotated, medium, or actually quite well? And then you're essentially done already. You will also be then able to say which online resources you used for it, where you went to make that decision, and that's it. If you have it may take only 20, 30 seconds. If you have a few more seconds while you're standing at the bus, waiting for a bus stop, waiting for the bus to come, or while you're in some other waiting at the centrifuge, you can do 1 or 2 or 3 of these, and in this way help essentially the entire initiative already to move forward by getting a broader information base on the challenge that we are facing. What we want to do then is, in fact, not to ask the question, what's the solution to the whole thing? Again, each of us may have intuitively a hunch. We may believe the solution is, and now you fill in your favorite technology, which possibly is in your laboratory. But instead of saying it like that would get us possibly also onto a very in a very good debating position, and we may bang our heads against each other. We would rather like to, before we start that, ask the question, how do we actually find a solution? So what is the best platform to start negotiating and discussing and weighing things against each other? So rather than saying, no, my tool is the best, let's think about what would be the best way to find, in fact, the answer of how we should move forward. So it's a bit on the meta level, but I think that's necessary, or we think that's necessary in order to identify the best approach forward. So it's not a top-down initiative. It's not me being the commander and saying now everybody should obey me. Not at all. It's the idea is let's come together and put all our thoughts and our awareness together and think about what is the best way forward. Cool. So that's why we, and now the we isn't just Georg and me, but it's also Tianan and a number of other scientists around the globe, in fact. It involves scientists from China, from Europe, from the United States, to come together and make the seed, the initial seed of this Understudied Proteins initiative, where our ground today is one element, and the survey is another element, and the third element is in fact that we are planning to do a workshop somewhere later this year, where we would like to come together and discuss, ideally in person or in some online format, or in some joint mixed format, how to proceed into the future on that. So this survey is not just giving us answers on these 3 questions, but actually it should fulfill also another very important role. And that is, we would love to use your information, your annotation of what you think is a well or poorly annotated protein, in order to train artificial intelligence to do the same statement, so that we can expand from the human annotation to from the few proteins that humans annotate to all the many proteins that are known in the databases. And we can keep doing this as time goes by. So we can actually look how the challenge changes over time, possibly as a result of our activities. And we can measure which activity, my favorite tool, your favorite tool, has the biggest impact on it, or which may actually, even though it's no very verbal person standing up for it, has in effect very little changing capability built in. And that could allow us to reallocate resources based on scientific evidence. And that would be, I think that would be possibly even unheard of in science. But that's this only works if we have enough human-based annotations. So that's again, please invest the minute or 2 or 5 minutes into going and helping us to assign the proteins. It's not done if I am just doing it all by myself, because then I would of course introduce all my personal views on it, and that would lead to a bias. We need to have a broad participation here. So do a few, you don't have to spend the entire weekend on doing it. Rather tell friends and colleagues with a scientific background, not maybe your grandparents, unless they are also protein scientists or very interested in this direction, to annotate a few proteins. Good. So with this, we would like to come to the workshop then, and report kind of like at the workshop the outcome of the survey, and discuss this annotation score that we hope to develop on the back of the survey. And we identified also already kind of like at least 6 areas where we think we need to discuss what to do. It's about the data generation, which technologies should generate what type of data, how the results should be disseminated. You mentioned already the influence of conditions and models chosen. We're going to have data integration issues, because now if we have loads of data from lots of different approaches, how do we integrate all this data? We want to monitor change, so not just generate hot air and activity, but we want to ideally most efficiently and effectively move from the current situation to an improved situation, fewer unannotated proteins and a broader study base. So widen the base of proteins that are making their way into research laboratories. And we have, of course, overall, an important element is quality control, because our data should be, like, of course, it should be good data, but good data needs to have an error margin attached to it. And there are some techniques that inherently, or that have this already designed into its proceedings. And there are other techniques actually that don't. And like, as an example, if you do expression level analysis, no proteomic based identification and quantification of proteins, of course, we have error assessment there. On the identification, we have error assessment, for example, through the target decoy approach. On the quantification, we have error assessment. Now we always have can measure CV values through replica. So it's very well controlled as a method. Now another method, and I don't want to make the method bad by this, I just want to give it as an example so you see that even though scientific methods can be very successful without, they do exist also without. So if you do protein co-purification, no IP-based, you identify things that come along with bait protein, but there's no error assessment there. You don't know what you miss. You don't know what you see. Is that now really an intactor or not? Is it a bait intactor? Is it like a bead intactor because you're having the bait immobilized on some beads? So there's uncertainty attached to this. And currently this data is going more or less uncontrolled into databases, and in that way kind of like also polluting the databases with interactions that aren't real. Now, no method is error-free, that's for sure, at least no scientific method. Of course, religious methods may be error-free by definition, by divine inspiration, but when we do science, we always have an error, and I think it's very important to be able to assess that error. Good. Okay, so these are areas that we think already, that we are aware of already, that need to be discussed in the workshop, and maybe you have other ideas what also should be added to these discussions. And there's a web link there where you can keep yourself updated on what's happening. Good. Of course, the Understudied Protein Initiative is not the only initiative in the world that that tries to achieve something, and in that wider space of proteins, protein functions, genes, and gene functions, there are a number of other initiatives, and we have placed them here to name them, so you're aware that there are other initiatives going on. The Structured Genomics Consortium, for example, that tries to solve a structure for every protein. Enzyme Function Initiative, the already mentioned Eliminating the Druggable Genome program, Open Targets, next CP50, and upcoming is the Human Proteome Project Grant Project, HPP, Human Proteome Project, isn't it? HPP? Tiananan, save me.
Yes, Human Proteome Project, the Grand Challenge Project.
Okay, grand. Good. So I just stumbled over the double project. Anyway, so all these have their specific focuses, and while they relate to the understudied protein challenge, they don't directly target it, and that's where we want to make a difference. Good. What we think, and this is now our hypothesis, is that proteomics has a unique contribution potential here, because ultimately when we are speaking about the understudied proteins, so probably studying the proteins is the most direct way to fill in functional annotation that will then help other scientists to further improve on this basis. And maybe this is an important consideration already. We are not trying to solve every aspect of every protein. So this is not what the initiative is about. It's about delivering enough information so that the protein becomes tractable to more specialized scientists to follow up with this protein with further detailed studies. And the question, what is required to give this minimal information, is one that we try to solve also through the survey. So access. And there are many proteomics techniques that are super powerful in giving some specific information on functional aspects of proteins. And because Georg and I have our own research, in fact, going in this direction, we want to present you two completely different tools, just to show you kind of like two pieces of the overall canvas that could help in order to assess proteins and provide some annotation. And that's the point where Georg will focus on one emerging, I would say, newish technology, and that is that of predicting protein function based on covariation and co-regulation.
Yeah, so protein covariation analysis is one of the things that we're trying to do in our lab, and it's we started doing this when I was a postdoc in Yuri's group. A few years ago, we published a database called Proteome HD, which is a catalog of protein abundance changes in response to biological perturbations, things like treating cells with growth factors, inducing differentiations, or treating them with drugs, etc. And we made this database purely by reprocessing publicly available proteomics data, although nowadays, with the availability of high throughput proteomics setups, it's quite feasible to actually gather even very large data sets from scratch. And in principle, though, it's always the same approach in which you then decipher function, which is by covariation analysis. So down here, you see a set of example proteins and their expression changes across 15 different perturbation experiments. And you see that they are selected to be part of two different groups. One group that has functions in oxidative phosphorylation, and they all have a similar characteristic pattern, and the next one is involved in ribosomal RNA processing in blue. I think you can appreciate that they have very different ways to responding to these perturbations. So if you can capture these patterns, for example, by correlation analysis or machine learning, even better, and compare them, then you have a way to group proteins together that have similar functions, and more importantly, to associate understudied proteins to proteins that are well studied, and thereby attributing a possible or predicting a possible biological role for them. And on the next slide, you'll see that we made a map out of this, which we call the human co-regulation or covariation map. In this case, every dot here is a protein, and it's plotted with a t-SNE algorithm. So that means the proximity between these proteins simply reflects how similar they respond to perturbations. We could also plot it as a network, but with such many proteins and many interactions, that looks more like a hairball because you have all these links. So we find this kind of proximity map to be a better solution. And on the next slide, you can see that even though this map is based purely on covariation, it does correspond partially, at least, to the physical location of the cell. So, for example, you have proteins from the nucleolus at the bottom, all grouping together because they have similar functions, similar expression patterns, and so on. And one thing that we noticed that was unexpected, among many other things here, was that the protein called PEX11, which is a well-known peroxisomal protein, was strongly co-regulated with mitochondrial proteins, especially with proteins from the respiratory chain. And so we thought perhaps this means there is an understudied function of PEX11 in mitochondria, or somehow related to mitochondria. And so we teamed up with Michael Schrader from the University of Exeter, who is a specialist in peroxisome biology, and he overexpressed PEX11 in human cells and then stained peroxisomes and mitochondria. And what he saw is that when you do that, peroxisomes, which are the green dots in this figure on the right, when you overexpress PEX11, they start to form protrusions that sort of probe the intracellular space and then reach out or getting in contact mitochondrial surfaces and essentially start forming a new organelle interface between peroxisomes and mitochondria. And we don't know yet for sure what this is about, but most likely we think it's about antioxidant protection, because if you upregulate mitochondria, that means, or if you produce more ATP more, you will also produce more oxygen radicals, and peroxisomes are full of antioxidant molecules. So it would make sense to also upregulate a process that will help protect the cell against oxidative stress. And so in this case, we've discovered a new organelle interface, but there isn't actually a physical interaction between PEX11 and mitochondria, although there is most likely a functional one. And that's just a quick overview of what protein covariation can do, and then I hand back to Yuri.
Great, thanks, Georg. And I would like to add, there's two very important things to take out of this. First, in fact, now we were speaking about understudied proteins, and here we have PAX11 beta. Of course, it has been studied before. There is knowledge that it is in peroxisomes. And yet we find through an unbiased investigation of protein function, we find new aspects of this function. And there will be many proteins, even among those that are already studied, that have alternative functions, sometimes referred to as moonlighting. And this is also an element that will come out of larger scale, unbiased, essentially discovery-driven approaches. So not based on prior perception, on hypothesizing and looking only at a very narrow thing, but keeping the view open, and so that will reveal new aspects of biology that so far remained hidden. And the other aspect is specifically on this method. No, this is a tool. This co-regulation analysis gives you things that are functionally linked, but not necessarily physically. They may be physically linked as well, but they don't have to be, as is in this case. PAX11 beta was found, in fact, co-regulated together with ATP synthase. No, so that's ATP synthase is in the inner mitochondrial membrane. It's not even the outer mitochondria, so it cannot physically interact with PAX11 beta. However, functionally they are linked, because when ATP synthase activity goes up, as Georg said, oxidative damage goes up as well, and PAX11 beta probably prevents or protects the cell from this oxidative damage by getting the peroxisome hooked up with the mitochondria. Now, the other technique that we wanted to kind of advertise to you is something that's completely different, and that is cross-linking mass spectrometry, which
Hi, Yuri, can I ask a question? Since there are so many technologies and a lot of new things I heard from you. So just now you mentioned a physical interaction between two proteins, and there is also an association, and also a causal or functional. They have a causal link, but they are not directly physical linking. So through this covariation analysis, do you think what level of evidence can we obtain from this type of analysis? Is it still association-based, or do you think you can determine the causality between two proteins, or between a protein and a function?
Well, what you do get is this information that two things, two proteins, tend to be, because it's statistical, tend to be needed together by the cell. And why does the cell need them together? It doesn't say. We only know they tend to be needed, both of them. And here we have an example where there's definitely no physical link between ATP synthase and PAX11 beta, but both tend to be needed together by the cell. So it doesn't just upregulate under one biological condition ATP synthase, it also upregulates PAX11 beta, probably because of the link of oxidative damage caused by one and oxidation protection by the other. So it's a functional link that is revealed by covariation, but not association in the sense of physical association. And the functional linking even isn't in the same process, but it is, the cell isn't served well if it just gets more energy, but at the cost of oxidation damage. It wants to be protected from the damage, and then it also upregulates mechanisms that help protecting from this oxidation damage.
Could I just add something to that? In transcriptomics data, people have already started looking at causal interactions, so regulatory networks, by trying to predict which transcription factors correspond with the upregulation of certain mRNAs, for instance. And the way that works is that you're trying to essentially explain if the variation in the target can be explained by the variation in expression of the transcription factor, for example, rather than the other way around. So that way you could, in principle, get directional information or causal information. I think that hasn't been done in proteomics yet, but I might be wrong. That would be very interesting to do. I know, however, that with the transcriptomics data, the accuracy of these data is often still quite poor. So I think for yeast, the networks that were made a few years ago, they had an accuracy of maybe 50% or so. But no doubt this will improve with more data and better algorithms.
Yeah, this is actually what we discussed last month in Cancun in the HPP Grand Challenge project. We want to perturb a protein by genetic CRISPR or by perturbation by a compound or antibody, and then we see the consequences of the many proteins, and to determine what proteins is a consequence of a protein, another perturbation of another protein, or another ETM of a protein.
Yeah,
so I think the project
for yeast, for yeast, the knockout library has already been measured. Markus Ralser, who's also part of the initiative, he has measured 5,000 yeast mutants for their protein expression level.
Yeah, that's very cool. But the human proteome project is study human. So we want to borrow those methodology to study human cells, and that is more expensive. Takes longer time.
But indeed.
Yeah. But the philosophy is the same. The philosophy of the methodology is the same.
Yeah,
very cool.
What one can maybe learn already from the yeast data is what type of information can be gleaned from such a study. What can you actually, indeed, no, I mean, nobody can predict exactly how informative in what directions this data will be. And as it is a lot easier in yeast and has in fact been done, but I think it hasn't been published yet, then one can get at least some first ideas. And when one wants to do a large project, it requires a lot of funding. You need to have also very solid arguments in place. Not only that it can be done, but also that it should be done because there's a gain of information, or there's benefit in it. So having smaller species, smaller scale investigation to build on may be very productive there.
Yeah, true. That's a proof of principle. But in the HPP, the Grand Challenge project, we planning a project for probably 10 years. Since during the past 10 years, we just characterized up to 90% of the protein. They are identifiable. But now next phase, we want to move to study the functions, understand the function of every protein that we can identify. Yeah, we can discuss later. I think there's a very great synergy between these two initiatives, and also with the UniProt. In UniProt, they have a lot of text information about every protein, which also mentioned just now. And we want more people, especially the audience from China, to participate in this survey. And based on the survey from manual inspection, probably we can develop an algorithm to automatically do that for the rest of the protein which have not been manually inspected.
I think even if we only achieve that, then we have achieved something very big already. Which is, I think it's quite exciting that, like, we don't want to build pyramids in Egypt, which may take a lifetime to build. We have something that, in fact, only requires many participants for a very short time, minutes. No, it's not even hours, days, weeks, and months. Just a few minutes of your very precious time, and instead of playing some online game for 5 minutes, play this online scientific initiative game and bring humanity forward a little bit.
You know, Michael Snyder, he had a very good experiment. every time he attend the HuB meeting, there's about 1,000 people joined. And then he will have a booth and encourage people to donate their biological samples, blood, urine, and stool. And they do this every year. And after a few years, there are maybe a few hundred participants. They donate every year, and they collect those samples and analyze the multiomics, including proteomics. So this year, I'm very lucky. I think we will have a lot of physical meetings. So we will have AO HuB, we have HuB, we have ASMS, we have Sing HuB, which I'm also organizing. I think we may consider set up some booths and put some small gift. I encourage people walking by to just fill a game with the iPad and to a few minutes fill those tests.
Yeah.
Probably will be
maybe also at the when it's going to the break time, when it says, listen guys, the break isn't now, so coffee is going to be there, but before you stand up, just for 1 minute, go to this website and annotate one protein.
Yeah.
That's it. And once they have seen how easy it is, maybe they do another one when they come back after break, before the talk starts, or maybe they sit in a talk which isn't hitting their interest, and then they are spending and annotating 2 more proteins. And that's all it takes. You know, as I said, we don't expect anybody to sit there and annotate the entire human genome. Not needed. Just a few proteins. Cool.
Yeah.
But let me, before we discuss more, push one complementary to this large-scale protein expression experiment that gives these functional connections, give a different technology, which is giving you not the functional links, but physical links, so direct physical readouts. And of course, our hypothesis, or the hypothesis of structural biology, is that if you know that 2 things are interacting, then probably they depend on each other for their function. And that technology is called cross-linking mass spectrometry. Just very quickly, there one uses chemicals that are bifunctional, and you add them to proteins so that they do react with both sides and generate a new bridge in the protein, which then connects parts of the proteins that are in the native folded protein close by. And if we then do normal proteomics, digesting with trypsin, analyzing by mass spectrometry, doing database search, we come back to an information of both how the protein, or some information about how the protein is folded. Of course, it isn't giving you the atomic resolution here, but just some proximities in the protein and between the proteins. And based on this, one can then build models. And this is here the example of a purified protein complex. But in fact, you don't need to purify the proteins. You can do the very same thing with the entire cell. So you can add the cross-linkers to the entire cell. Of course, the cross-linkers have to pass the membrane, which not all cross-linkers do, but some do. And then you do have a very complex mixture. You have a lot of peptides. You need to do a lot of analysis, and then you get a network out. And of course, again, on this, error estimation is very, very important, but there are some papers on this out already. So that exists as a proteomic technology. We do have here some error estimation. And we did this in a bacterium, Bacillus subtilis, and this is how our first network looked like. And what was particularly interesting for us was the blue dots here, and those are proteins of unknown function, which was surprising to us because, of course, we are seeing mostly just the abundant proteins right now. In Bacillus subtilis, there's over 4,000 protein coding genes, almost 4,200. And of course, there's many more interactions than the ones that we have detected here. So this is not exhaustive analysis yet. But even though we're only seeing the tip of the iceberg, we see a lot of uncharacterized proteins in these interactions. And that gives us, for each of those interactions, it tells us a hypothesis. No? And that can be probed. And I want to show you at the example of pyruvate dehydrogenase, which is a very well-known metabolic complex, very well studied in many, many species. And we find here an uncharacterized protein is attached to it. That's quite surprising how such a well-known complex has here a new factor. And the reason why we see it is in fact linked to the cross-linking in cells. Because if we lyse the cells and then do size fusion chromatography, we see that this protein, which we now here call pyruvate dehydrogenase inhibitor, and I'm going to show you in a second why we renamed this uncharacterized protein to PDHI. You see that in untreated cells, it doesn't co-migrate with PDH, but once we cross-link, it does co-migrate. So lysing cells leads to the loss of some interactions. And of course, it doesn't know some interactions survive the lysis, and those are the interactions that tend to be studied so far. But if we cross-link first, then we stabilize these interactions. And cross-linking mass spectrometry, based on in-cell cross-linking, then can detect these so far hidden interactions. Now, knowing that these proteins interact, we can use, for example, AlphaFold to predict the structure. And here we predicted this protein. That's the previous name, Webniar, as an uncharacterized protein, same as PDHI. We can predict the structure for the two. This is a way of assessing a structural model coming out of AlphaFold multimer. When it's blue, it means it's good. It's low positional error. So that's just a quick introduction to the scoring mode here. And we see the cross-link not only from Webniar to PDHA, but also to PDHB. And if we predict that one, in fact, there you have an example for a poor model. This is red, high positional error. So that means that actually AlphaFold fails to predict a good quality model for these two proteins, although we know they do interact in cells because they're cross-linked. But now that we know that all three proteins seem to be interacting, we can throw all three of them together in AlphaFold, and voila, there you go. AlphaFold presents a model which it actually believes to be of high quality, low positional error. And now we have, based on in-cell cross-linking mass spectrometry, that was low resolution structural data. We just see, okay, this protein, that protein somehow together like this. But now with AlphaFold, we get a pseudo-atomic model. Now we know kind of like how things might be falling together. It's not proven, but they might be looking like this. What is interesting, so the model fits also the crosslinks. We didn't put the structural information of the crosslinks in there. This model does fit. And it was, by the way, it was the 7th ranked model by AlphaFold. It wasn't the top ranked model. The top ranked model, in fact, did not fulfill the crosslinks. So crosslinks, they help also in model selection. But you see that this protein, this new protein, sits right on top of a little bit of red here. And this little bit of red in the background is, in fact, pyruvate. That's the binding pocket of pyruvate. And this so far uncharacterized protein binds right on top of it, presumably blocking the access. And now here you have the tyrosine penetrating into a hydrophobic pocket that normally pyruvate enters through. So it can probably not enter when this Webniar/PDHI is bound. And in fact, when we overexpress the protein and grow the bacillus on pyruvate as a carbon source, then we see no growth over time, that the overexpressing cells, they don't grow very well, whereas the other ones grow fine. So the protein seems to slow down, and that is pyruvate specific. If you feed them on glucose, it doesn't matter that we overexpress this protein. We did one more thing, and that is knowing now from the structure that this amino acid seems to be important, and also that one, this arginine here, it makes loads of hydrogen bridges. We mutated this arginine and we mutated this tyrosine to alanines, and both mutations, in fact, abrogated this effect and thereby validating the structural model. And that's just the take-home message of this part of the talk that I want to give to you, is that this cross-linking mass spectrometry paired with AlphaFold gives actually a super shortcut into structural biology and into functionally annotating proteins at quite a high level. Of course, this is not the end of functionally studying this protein. It's just the beginning, but it's a very solid beginning. Now we don't know how it's regulated. Why is this? When is this protein doing its function? When is it not? And so on. So all that is yet there's still PhD theses that can be written on this protein, but now it's very clear who may be interested in that. People that are interested in PDH may now study this specific uncharacterized protein. So it has found a home in biology. Great. And we love that many proteins receive these kind of initial functional knowledge boost so that that then further studies can be initiated in a targeted way. And no, that's which leads us now to the final slide. This is again here. If you want to do the survey, please do take a picture of that and go there and do a few. We are on our way in this initiative. Well, thanks to this presentation, thanks to this invitation, and thanks to your participation.
Thank you. Jury, can you go back to the previous slides? I have some question. So you combine a cross-link and AlphaFold. This is very cool. Some conventional structural biologists may ask, what's the resolution of the structure that you can resolve with this computational approach? It's not based on cryo-EM or those X-rays, but based on computational. And this is also related to another question I want to ask about the understudied protein initiative. We discussed in Cancun, people also ask the same question: what's the function? What's the function of a protein? Even for p53, is this really well studied? Is this protein really exhaustively studied? Or there are so many levels of evidence. Some conventional biochemists, they prefer to have a very detailed mechanistic function of a protein. And like some structural biologists, they want to see at a near atom level structure of a protein. But for us, for me, we are studying proteomics. Normally we will study protein association, or sometimes we establish a causality of different protein functions. So there are different levels of functions, different levels of how you define study. So in particular, in this case, we provide some evidence for this protein structure. So how do you?
Yeah. So it's two very important questions. Let me take the smaller one first, and that is like, what is the confidence in this model? I would disagree. It also bases on crystallographic data because the algorithms learned, of course, from the solved structures. I mean, they didn't get their knowledge of how proteins should look like out of thin air. They base on a lot of data, but not specifically on this complex, of course. So we use artificial intelligence that looked at thousands of solved structures plus evolutionary information, multiple sequence alignments, that together comes back with a prediction. And the seventh model, in fact, just to emphasize that again, the rank 7 model was the one that fit our crosslinks. The few crosslinks, that certainly doesn't allow us atomic resolution. Like a few crosslinks here just says, well, this protein sits somewhere around here. It could sit in many different ways. But thanks to AlphaFold, we come back with a very detailed model. And this is nothing but a model. It may be totally wrong. As it turns out, AlphaFold not only spits out models, it spits also out a number of scores. And assessment so far speaks for that these scores are actually, even though we don't really understand how, but they are quite reliable. That doesn't mean that they are never wrong. Again, we are doing science. It's only kind of like chance of being right high or chance of being right low. And if the scores say it looks pretty good, often it actually is pretty good. But we don't know if the case that we're looking at is a case where the score is right or wrong. So it's very important that this is just a model that then allows us to move forward. And in this case, the model told us this tyrosine is super essential. It's very important because it inserts into hydrophobic pocket. And then we do what structural biologists do. You do a point mutant, and you ask, well, does the point mutant have the effect you would expect? And it did. And that, of course, we cannot, that doesn't prove that every side chain in this model here is correctly positioned, not at all. But it lends very large credibility to this model because this tyrosine was essential. This arginine also was essential. So through these point mutants, you bestow credibility to the model. And now to the other part, there are many definitions. There's possibly as many definitions to function as there are scientists, or maybe even more definitions to function, and they are all okay. I don't want to enforce any definition of function onto anybody, but for us as an initiative, I think our aim needs to be clear, and our aim cannot. Well, maybe it could, but it would be a very unrealistic aim to satisfy every definition of function for every protein. I don't think that's the endpoint for the initiative. Our endpoint is much more humble, much more humble. It is to give enough idea about the function so that others feel, oh yes, this is a protein for me, and now I want to spend the rest of my life on this protein. That's grand, because I think there's no endpoint in studying any protein, probably. There's always something more you can find out.
Yeah, this is very cool. So this is actually in complementary to the HPV Grand Challenge project, where we want to establish a definition for a function to start those Grand Challenge project. You know, during the past 10 years, the HPP characterized about 90% of the human proteome, the non-protein genes. I mean, every protein for each gene, and they have different levels of so-called identified proteins. There are protein evidence level 1, level 2, 3, 4. And now in Cancun, we discussed about to define evidence level. So evidence of function. So function level evidence 1, 2, 3, 4. And so the highest level probably by a very rigorous mechanistical study. And the least level of evidence probably by computational prediction. As you said, we normally, a model have a very high degree of precision, like AlphaFold seems higher than 90% or 95%. But in reality, for a particular protein or protein function, the real error rate is somehow very difficult to determine. So, so I think the function level definition will be very good complementary to this UPI initiative. A lot of people ask me, ask Joey, what do you plan to do in the UPI? So very unfortunately, he could not come, but I think it's very good you have a workshop. We can invite more people from other communities, other initiatives to join us in this.
Shall I stop sharing? Then we see each other larger on the screen.
Yeah. Yeah, cool. Thank you. Now, thanks for your presentation. And then we have a panel discussion about this UPI and how we can proceed. I know there will be a workshop later, maybe online or in person, and we should encourage people to participate our survey. I have another question. I have a colleague, actually I have several colleagues at Westlake University who is from School of Engineering. They are computer scientists. So they are working on text mining. They asked me, what can we do for the life sciences? When I read your PPT and the paper, I think since individuals can build the survey, can we train a machine to do that by having some real experts of who knows a lot of biology of every proteins. Instead of have 100 people working, every people working for 1 minute, we may have 1 people working for 100 minutes to build up an annotated training set. So I know in the natural language processing field, they can learn how a real expert read a text and make a conclusion. And all this process can be simulated by computers. If you have 100 people working for 1 minute, they may have variability. So some may be more experienced than the others. Have you thought about this?
Yeah, indeed. You want to, so I'm not talking the entire time.
Yeah, no, I mean, this is one of the things we've discussed about the server, that on the one hand, it's good if we get as many different opinions and that many different people as possible, because it would allow us to capture a bigger variety of what people actually understand when they say function. On the other hand, of course, it would be good if we could somehow also distinguish or perhaps weight answers based on the experience or based on the amount of work someone has put in. But I think we concluded that that would be very difficult for us to do. But I guess we didn't consider text mining for that.
No, but let's, I mean, there's also two different questions at least here. So let's come to the text question mining second, and first the annotation aspect. So one thing that is important to keep in mind is, of course, there's on every studied protein, there is somebody who is like the world authority on that protein. But we don't need that person, because if this protein is like, by anybody's definition, quite well studied, well, we put it in the well studied box and that's it. No, because we are not trying to collate all the knowledge about this protein. We just want to know in which box to place this protein. And the very poorly studied proteins, well, I mean, they are very poorly studied, so there is no expert. You just look at it and go like, well, I can't find anything about this protein. Well, that means it's very poorly studied. So let's put it in the poorly studied box. And once we have ideally all 20,000 human proteins put in these boxes, then that would be our training set. And that's indeed when computational experts are needed, because then we would love to take this training data and take all the different resources in the internet that we can find and bring them together and learn what is it that people are using. What is it that makes to people a protein well, decently studied, or what is missing that it is poorly studied? No, because, I mean, now just hypothesizing, maybe cellular localization is a very poor indicator of function. So it would be not well invested funding to say, let's measure the cellular localization of every protein. Or maybe inverse, maybe it's a very strong indicator of function, and then we should invest into measuring. Now, we can debate whether it is or not, according to our personal beliefs, but if we have that data coming out of all these annotations in a systematic assessment, well, then we have kind of like an answer to that. And I think text mining could help a lot. I mean, there's now this machine learning GPT engine that can write apparently really decent essays. The problem is the score, whether it's good or not. But at least it's a decent essay it can write. And I could imagine, I can recall that there was once the initiative of, in fact, doing this collation of what's all known about each protein. So then you would need the world leading authority on every individual protein to collate like all their knowledge and write that on one protein. But given machine learning, maybe we can do that at least as a like model annotation. We know it's maybe not absolutely correct, but even if humans were to do it, it wouldn't be absolutely correct. No, because we also have wrong data around and people overlook stuff and so on. So never it's absolute correct. And if we put the machine generated annotation clearly on the top, then people know. Maybe it's right, maybe it isn't, but at least it is there. And you don't have to go and search the internet yourself. So it shortcuts a lot of activity. So I think that would be cool. So there's two. One is once we have our annotation data as the training data, then finding the links to all the information around the internet and weighing them, what makes a protein annotated well or studied well or not, and using like this machine learning model that has been developed now, or its successors in the future, in order to provide kind of like short abstracts on every protein that collate what is all the knowledge that is around in a nutshell. When I want to know about a protein, I typically go to UniProt and read what does UniProt tell me about it, like just very quick, of course. And they do, no, that's manually generated, a lot of effort and very, I mean, once they've written something, it's not updated very often. And if you were to do this automatic, then you could run this annotation, I don't know, once a month and keep it up to date, no?
Yeah. So then I have another next question. You just now you mentioned the endpoint will be some level of annotation for every protein, based on maybe computational weight approach or based on collection of information from the literature. So my question is, what level of granularity do we expect for this information for every protein? You can say this protein is related to transcription or related to protein synthesis, or you can say this protein has relationship to pancreatic cancer, or it can be a target for some compound. These are different levels, and people, biologists or clinicians from different fields, they look at different aspects of protein. Should we define several minimum level of information that we should achieve in this initiative for every protein?
I think that's very well observed, Jianan. So for the, in fact, there are some of the initiatives that we named that have kind of like different aspects of the functional space as a target. And when we speak about druggable
druggability, yeah.
No, then they specifically hunt after the small molecule binding aspect. To me, I would, but that's not my subjective. But in fact, some aspect of this should come out of the survey again. No, because that's where we ask for what do people understand under function. Because it comes again out of when they sort these proteins into boxes. And the level of granularity also, what does it require for a protein to become tangible? Now, when is it studyable by dedicated researchers that want to study this specific protein rather than just like thousands of proteins at the same time? Now, when does it make that transition out of the omics field into the one molecule at a time field? And I don't have a definition for that right now, but we have set the survey in a way that we should be able to get the answer out from the survey. That's cool. Why it has such a paramount importance, in fact.
Yeah, very cool. So a question for George. You mentioned a covariance analysis. I think that's really cool. You have more than 10 perturbations for the system, for the cell, and you monitor the proteome, and you see some protein blue, some in red. They have different clusters. But how many perturbations can you perform for this system? Every perturbation, they only affect a subset of a proteome. Do we have enough perturbation to perturb all the proteome, the entire proteome, in particular those understudied proteins, so that we can scale this up? Otherwise, let's say most of the common, drugs for cancer cells, they're going to kill the cancer cell. They will perturb metabolism, apoptosis, and so on. And some essential pathway probably they can hardly be perturbed, and then give you insight of those understudied proteins.
Yeah. So one of the interesting things we've seen with this type of analysis is that often the perturbation, or generally perturbation, does not have to be related to the function of a particular protein that you're studying at all, because for instance, in Proteome HD, we're looking at SILAC data, so we have very high quantitative accuracy, and we can see small changes of plus minus 20–30% or so of an abundance. And that level of change is often achieved just as secondary effect, or just as a so it's rather easy to observe or quantify those small changes, and so you can quickly cover nearly every protein, I think, if you have sufficient accuracy. In general, the more data you have, the lower the accuracy of each quantitation can be, because as you accumulate more data, you can still see pattern even if the resolution is lower. So I think so far in the human proteome, the map that we have so far covers only in that Nature Biotech paper covered 5,000 proteins. And the reason we had to exclude the others is not that they weren't perturbed, but because we couldn't detect them in sufficiently well enough experiments. But that was more a question of scale of the data itself. So we're now working on larger data sets to change that.
Yeah, this is something we can probably also collaborate with the Target 2035. They have a library of compounds which can be used to perturb different system cells, because not every cell expresses every protein. And in a Grand Challenge project, we also started a Westlake pilot project. We are working on triple negative breast cancer cells. We have accumulated over 6,000 perturbed proteomes. The breast cancer cell line treated with different drugs used in clinic and combinations, different time points. So I will be very keen to try your algorithm after we this data set ready. So another question, maybe to Jory. What do we have grant to support this initiative? So during the past half a year after our paper is published, we have some promotion of this initiative in China, in Chinese. And many people interested. I also talked to some colleagues in the States, and they are also expressed interest. But they asked me how we can participate. Is there a grant to support this? So I say, tell them you fill the survey first. So they probably spend 1 minute to fill the survey. But then what's next? Do you have any concrete plan?
Yes. Actually, it's one of the large weaknesses of proteomics to have high dreams and little data to support those dreams. And funding agencies, they are very happy to give money. They actually give a lot of money to science, but they want to have hard evidence. So genomics is very successful in securing very large grants based on hard evidence. And this is what we have to provide as a proteomics field. So we have to understand that we first have to put in some work, and then we get access to the cash, kind of, when we have very clearly defined objectives and provide tangible measures of progress. And this we don't have right now. Now we can say, well, there's all these uncharacterized proteins, but I don't even know what an uncharacterized protein is. And we're going to do lots of experiments, but we don't even know how these experiments will change the situation. So consequently, in the moment, all we have is a gut feeling. And on a gut feeling, you don't get funding. So we have spoken a lot to funding agencies, in fact, and one funding agency, the Wellcome Trust, is involved. Now they are part of this initiative, and they are willing to give us, well, they have multiple billions of endowment. They are not willing to give us any of these billions. They don't even give us $1 million right now. And they give us the money to do the meeting. No, but it is something, and they paid the publication fees, which was also something. No, it's no, publishing Nature Biotech isn't a cheap thing, but no, they upped their involvement as we provide evidence. So again, in fact, your answer is right. Fill the damn survey, because the survey is our entry ticket. Like if we have very little annotation of proteins through that, then we don't have a training group, then we can't make an automatic assessment later on, we can't monitor progress, we can't have a definition of what is the right granularity, we don't know what actually function is to biologists versus omics researchers. So we lack all our bases, and if we lack the bases, I mean, we can keep going to funding agencies and they will keep telling us exactly the same thing as you don't have a case.
So
Yeah.
I would say this is part of what you come out of the workshop, and it will be very important to have a very simple definition of function and what we want to achieve. So for example, if we find that if a protein needs to have at least 10 known associations or so to overcome this streetlight effect, so that once something has 10 associations, then people start picking it up from screens and so on, then it would be an easy case to say we need to find 10 protein-protein association for every single human protein, and that will be the start to overcome this challenge. But I think it needs to be something very simple and supported by evidence.
Yeah, that's very cool. So the same thing also happened for the Grand Challenge project. There are a lot of scientists from different countries. They agree that it's so important to get a function for every protein, but there's still not funding to support this initiative. And probably you also know that the next plot from Switzerland, actually they ceased the support for this annotation project. But the good news coming out also from Cancun is in China, Dr. Fu Chuhe from Beijing. So he led a team. We got a sizable grant for the proteome research, which is called a HuB, the human navigator of the proteomic navigator of the human proteome of the human body. It's a HuB. I can send you more information later. So this will be a multi-billion project for next 30 years. We will be funded by Chinese government, and they have a really interest. We have shown some evidence that is so important to develop proteomics, but it's not only for proteomics. It's also for other omics and also for clinicians. Because we need to help them have their research, like what they did in the genomics field. So it'll be very beneficial for every party that if there's a big initiative, they can join forces and to convince the agency from different countries to support those next generation biology, the omics plus AI. I will be very keen to continue more discussion with you. So we have a lot of audience online, but I don't see many questions from them. Probably this topic is too relatively general for them, and maybe all they are working on building the survey.
Hopefully, and well spent.
So maybe we can. Some of the audience they say they can you speak Chinese?
你好吗?
Still learning.
Again.
We still need to learn that.
There is space for improvement, but I have learned 你好.
Ni hao. Ni hao. Cool. So this seminar is also recorded, and maybe later we can add some subtitles in Chinese so that more audience from China can understand. And I also think about maybe we can translate some of those survey, or it's a very simple survey. If you need help, I can help you to translate into Chinese, and we can promote this survey in China. Because some of the students, when they see a lot of English words, they think they have a little bit of pressure. It's like yeah. So we can also possibly promote this in other Asian countries. I noticed Dr. Techno Yu is also online, and he's the Secretary General for HuB, Asia Pacific HuB, and he's very active. And we have a lot of scientists from Singapore, Malaysia, Korea, Japan, and in HuB also discussing how to promote an initiative. One thing we may do is to have a multiple language for the initiative, so that more people can participate. If you have this in German, probably more German students will join, or German clinicians will join. That's some of my idea. So we can better think about this. I look forward to workshop with you in the near future, either online or in person will be really cool. Or you may join the next HuB meeting in Korea or in China, and then we can show you around. And as I said just now, maybe you have a booth in a HuB conference. There are 1,000 people there. Probably 50% will fill in this survey. Okay.
That sounds great. Yeah.
Do you have any other comments?
Maybe one more Chinese word. 谢谢.
Oh, 谢谢. Thank you. Yeah, 谢谢.
Thank you.
Thank you. Okay, thanks a lot. We have 1 hour and 30 minutes right now. So thanks for your time and a very nice presentation. I'm very impressed by the street naughty story. I also talk about this different version in my presentation in China. I talked a lot to other clinicians, biologists, and many people are interested. And many biochemists, they are interested to contribute. They are not from proteomics, but they think they can also contribute to the proteins understudied. So I look forward to more discussing with you, and let's keep in touch.
Thank you.
Okay,
Thank you.
Bye-bye. Bye-bye, everyone.
Bye-bye. Have a nice day.